Welcome to Little English Studio, the channel where history, science, and human ingenuity come alive in clear and captivating English. Today we travel to the molecular world, to a single image that revolutionized biology, genetics, and our understanding of life itself. This is the story of Photo 51, an X-ray diffraction image of DNA that provided the key to unlocking the double helix, an image that changed the course of science and became a symbol of discovery, collaboration, and controversy. Join me as we explore how scientists captured this image, why it was so critical, the people behind it, and the profound impact it had on biology, medicine, and the way we think about life. The story of Photo 51 begins in the mid-20th century, a time when the structure of DNA, the molecule of heredity, remained one of biology's greatest mysteries. Scientists knew that DNA carried genetic information. They knew it contained 4 bases: adenine, thymine, cytosine, and guanine. But the arrangement of these bases, the way they formed a stable yet flexible molecule capable of storing and transmitting information, was unknown in 1952. Rosalind Franklin, a brilliant chemist and X-ray crystallographer working at King's College London, captured an image that would change everything using X-ray diffraction. Franklin directed powerful X-rays at crystallized fibers of DNA and recorded the patterns made when the X-rays scattered off the atoms in the molecule. Among these images, one in particular, known as Photo 51, showed a distinct X-shaped pattern, a clear indication of a helical structure. The image was remarkable not only for its clarity, but also for the subtle details it revealed. The spacing of the spots suggested a regular, repeating pattern, the dimensions hinted at 2 strands, and the diffraction pattern indicated the angle and pitch of the helix. Franklin's meticulous technique ensured that the DNA fibers were well prepared, hydrated, and aligned, allowing the X-rays to produce patterns that could be interpreted quantitatively. She measured the distances between diffraction spots, calculated angles, and noted the molecular symmetry, all of which contributed essential parameters for building a physical model of DNA. Yet Photo 51 did not exist in isolation. It was the product of years of careful experimentation, of understanding the chemistry of DNA, the behavior of fibers, and the physics of X-ray scattering. Franklin and her graduate student, Raymond Gosling, systematically prepared samples of A-form and B-form DNA, adjusted humidity, and recorded multiple diffraction patterns, documenting the molecular subtleties that others had missed. In London, the photograph sat quietly on a lab bench, but its influence would soon ripple across the Atlantic. In Cambridge, James Watson and Francis Crick were racing to determine the structure of DNA. Their approach relied on model building, but they needed accurate measurements to construct a correct helix. Through a combination of fortunate circumstances and contentious access to Franklin's data, including the examination of Photo 51 by Maurice Wilkins without Franklin's direct permission, Watson and Crick obtained the critical information that allowed them to deduce the double helix in 1953. The X-shaped diffraction pattern revealed the helical nature, the width of the molecule, and the spacing of the bases. These insights enabled Watson and Crick to propose a model in which 2 strands of DNA wound around each other, held together by specific base pairing, adenine with thymine, cytosine with guanine, a structure elegant in its simplicity, yet profound in its implications. The publication of the double helix in Nature in April 1953 immediately transformed biology. The double helix explained how genetic information could be copied, how mutations could occur, and how heredity functioned at the molecular level. Photo 51 was the hidden hero, the empirical evidence that made the theoretical model possible. Yet the story is also one of human complexity. Rosalind Franklin's contributions were underappreciated during her lifetime. She did not share in the 1962 Nobel Prize awarded to Watson, Crick, and Wilkins, in part because the Nobel is not awarded posthumously, and partly because the recognition of her critical experimental work came later. Today, Franklin is celebrated as a pioneer, a meticulous scientist whose brilliance and dedication were crucial to one of the most important discoveries of the 20th century. The technical brilliance of Photo 51 also deserves attention. X-ray diffraction relies on the wave nature of X-rays and the scattering caused by electrons in the molecule. The regularity of DNA fibers produced constructive interference patterns that encoded spatial information. Interpreting the diffraction pattern required understanding Fourier transforms, crystal symmetry, and molecular geometry. The X pattern of Photo 51 indicated a helical structure, the spacing of the horizontal layer lines revealed the helical pitch, and the distances between spots suggested the number of nucleotides per turn. Franklin's attention to these details meant that the data were not only beautiful images, but also quantitative blueprints for constructing a model. Beyond its scientific significance, Photo 51 symbolizes the nature of discovery, how observation, careful measurement, and interpretation converge to reveal truths hidden from the naked eye. It also highlights collaboration and the ethical dimensions of science. The debates over data sharing, recognition, and credit continue to resonate. The photograph became an icon, reproduced in textbooks, exhibitions, and documentaries, inspiring generations of scientists, highlighting how a single image, captured with patience, skill, and insight, can unlock mysteries of life itself. The impact of Photo 51 extended far beyond molecular biology. It laid the foundation for modern genetics, biotechnology, and medicine. Understanding the structure of DNA led to insights into genetic disorders. The development of DNA sequencing, forensic science, gene therapy, and countless applications that touch daily life. The elegant simplicity of the double helix, revealed by that diffraction pattern, demonstrates how form and function are intimately connected at the molecular level. Educators use Photo 51 to teach concepts of structure, inference, and the power of visual evidence in science. The story of Franklin, Watson, Crick, and Wilkins is also a cautionary tale about the human dimensions of scientific work, reminding us that recognition, fairness, and collaboration are as critical as technical skill. In recent years, Franklin's legacy has been increasingly acknowledged. Institutions, awards, and public awareness have honored her contribution, and Photo 51 remains a central artifact, a visual testament to both a scientific breakthrough and the people behind it. Viewing Photo 51 today, one sees not just a scattering of spots on film, but the blueprint of life, a visual code that would transform medicine, biology, and society. It is a reminder that science advances through observation, interpretation, persistence, and the courage to look closely at the details that others might overlook. And that was our journey into the story of Photo 51, the X-ray diffraction image that revealed the double helix of DNA and changed the world of biology forever. If you enjoyed this deep dive into science, discovery, and the human stories behind groundbreaking research, please like this video. Leave a comment telling us what amazed you most about Photo 51 and Rosalind Franklin's work. And subscribe to Little English Studio for more cinematic stories told in clear and beautiful English. Until next time, keep exploring, stay curious, and remember that sometimes a single photograph can change the way humanity sees itself.